Ya, no te
Ahí
escuchamos,
sí,
estás ahí, sí. Ahí
ahí
sí.
sí. ¿Se ve la pantalla?
Perfecto.
Ok. Bueno, muchas gracias a Sergio, a Adriana y María José por esta oportunidad de presentar en ACCDiS. Em, Muy importante para mí porque es una forma de tratar de reconectar igual con la comunidad científica en Chile. No, no es tan fácil. Así que muy agradecido a Sergio por la oportunidad de presentar este trabajo. Ahora voy a cambiar al inglés. Eh, bueno, esta presentación la he preparado en inglés, así que es más fácil para mí. Ok. Um, Today, I will talk about unpublished data that current ongoing investigation about the interplay between cholesterol metabolism and the endoplasmic reticulum remodeling via ER-Phagy. Oh, wait a second. There. And I will change to let's go. Okay. So ER-Phagy is the selected autophagy of the ER, endoplasmic reticulum, and is basically a process in which fragments of the ER are degraded in the lysosome via autophagosomes. And for this process to happen are essential a group of proteins called ER-Phagy receptors, which most of them are ER membrane proteins. And for example, there is this group of protein called FAM134 family proteins. And the main characteristic of these proteins is that they have an LC3 interacting region, which allows the engulfment of this portion of the ER to the phagophore, which then matures into an autophagosome, and then the ER content is delivered to the lysosomes, where it's degraded and the components are recycled back to the cytosol. So what is known about this process has been extensively studied the last 8-10 years. And what we know about this is that it's a primary response to nutrient stress, for example, amino acid starvation. It's induced by mTOR kinase inhibition, for example, using drugs like taurine-1 or many others targeting this important kinase that regulate metabolism. It's an ER quality control. There are a few examples that aggregates that are potentially toxic for the cell that accumulate in the lumen of the ER are targeted by this pathway, so then the cell can eliminate these toxic aggregates. Importantly, for physiology, this process is required for neuronal homeostasis. And for example, loss of function mutation on some ER-Phagy receptors in patients are associated with hereditary sensory and autonomic neuropathy, and this leading to, of course, linking to neurodegeneration, this ER-Phagy pathway. Recently, in vitro model using is also required for neurogenesis in vitro, and lately many groups have been trying to link ER-Phagy with innate immunity response, which is also very interesting because, of course, of many is linked to human diseases. So here during the last years, I've been focused in the study of these ER-Phagy receptor proteins. And there is already described near to 10 proteins that they do this pathway. We focus here in this family of proteins called FAM134. This is mainly 134B, FAM-B. And this FAM-B has two isoforms called B1 and B2. And the main characteristic of these proteins are that they are reticulum homology containing proteins, which means that these proteins are helps to shape the ER membrane. So this characteristic reticular form of the ER is basically driven by this kind of protein, these reticulum containing proteins. So the FAM-B has two hairpins, helical hairpins, here in the N-terminal, and this other hairpin here in the C-terminal part. And also important characteristic of this protein is amphipathic helices, and all these elements helps to shape the membrane in the ER. And there is a short isoform which lacks this N-terminal part and the first hairpin, only contain the second hairpin, and both, although they contain the LC3 interaction region that allows the targeting of ER to the autophagy pathway. And here there is a model that was published here in the lab, and here we can see again the two hairpins here in green and the amphipathic helices. And importantly here, that these C-terminal and N-terminal regions are disordered, and in one of the C-terminal disorder region is the LC3 interacting motif, which allows the targeting of the ER to autophagy. And another important characteristic of these proteins are that they form clusters on the membrane. So, for example, here in this molecular dynamic simulation, it was described that this the reticulum domain of these proteins is able to first to sense slightly curved part of the membrane, and then this represents like a sorting mechanism, and which many molecules can go together to this curved membrane, and this forms a cluster, strong cluster, which will generate even more positive membrane curvature. And this shaping of the ER is essential to then so that this portion of the ER is target to autophagosome. And this clustering event of these FAM molecules is relatively easy to detect in the confocal microscopy. And you see here in green the expression of FAM-B wild type, and you can see the ER network, and in some point of the ER are increased local concentration of the protein, like here, and this part of the proteins are positive to other ER shaping proteins like RIP5, and also recruit the autophagy machinery. So these clustering events occur in a specific part of the ER, which so far are still kind of a mystery. We don't know exactly in which specific part of the ER ER-Phagy is happening. We published with Adriana, we published that also these clusters contain ubiquitin, and then ubiquitin also helps to stabilize these clusters and boost the ER-Phagy pathway. So we were very interested into study how the content of these FAM-B clusters. We developed some tools to do that. Just briefly about this, we were able to force the generation of these clusters in the cell, like here, this cluster, using this tool that is bimolecular complementation affinity purification, which is basically that we fuse one molecule of FAM to the N-terminal part of the venous protein, the venous fluorescent protein, the other molecule of FAM to the C-terminal part of the venous protein, and we express these proteins in cells, and when the FAMs are coming together, this venous is reconstituted, and we can observe the fluorescence of the protein, but at the same time we can immunoprecipitate these clustering events using just normal GFP IP experiments. So this allows us to observe the protein in the cell, but also, for example, study this clustering and its content, but using mass spectrometry. So this is what we did. We characterize the interactome of these FAMB clusters in the ER membrane. And we describe in this paper that ubiquitination machinery was present, also autophagy machinery, of course, like these ATG8s. And also we were quite surprised, we found also many other reticulum proteins. And this, of course, makes a lot of sense because FAM recruit more ER-shaping proteins to bend the membrane, to generate curvature, and then this budding vesicle. So after this study, we were interested also what else was present in these FAM-B clusters. For our surprise, we found a lot of enzymes, ER enzymes, which are most of them are ER membrane enzymes that participate in lipid metabolism. Among them, many of them were related to cholesterol metabolism, here highlighted in red. And so just driven by curiosity, we started to ask if FAM ER-Phagy is connected to cholesterol metabolism. So many of these enzymes are part of the novel synthesis of cholesterol that happen on the ER membrane. For example, here we have one example, important example, SQLE, which is a rate-limiting enzyme of this de novo synthesis in the ER. Facilitate the first oxidation step of the esqualene, and then there are these two pathways that they both, the final product of these two pathways is the cholesterol. And as you can see, many of these enzymes here, like DHCR7 or 24, are in this pathway. So the simple question was, in this moment of this time of the project, is cholesterol able to modulate degradation of the ER via autophagy? And for that, I established an assay, the most simple one that I could develop in the lab to study how cholesterol might be regulating ER-Phagy. So the assay is very simple. There are commercially available serum that are lipid depleted. And so we are able to perform depletion of esterols in the cell. And we combine this special serum with inhibitors of de novo synthesis, like, for example, this inhibitor that inhibits here SQLE. So it's important to say that we are inhibiting both the novo synthesis and the cholesterol that can be provided in the serum. And also we use methylcyclodextrin to increase the sequestration of cholesterol and increase the depletion. Now, importantly, after the depletion, we are able to add back cholesterol, to supplement it back. So in that way, we first deplete, and then we add back cholesterol, and then we study more specifically the effects of cholesterol on ER-Phagy. So important for understand this assay is that many of these enzymes that are involved in the de novo synthesis are undergoes feedback regulation by cholesterol itself. This means when the cell sense lack of cholesterol, these enzymes are upregulated, and opposite, when the cells sense that the cells are okay with the levels of cholesterol, most of these enzymes are downregulated, and this happens at every level: transcriptional, post-transcriptional, and post-translational. For the purpose of this presentation, we focus here in the post-translational regulation, which means that when these enzymes sense, when the cell sense, especially the ER sense, that there is sufficient levels of cholesterol, all these enzymes are downregulated, meaning that they are degraded. And this is an example for SQL and many other of these enzymes. So this is the data supporting that our assay is working. So here we incubate the cells with normal FBS, and we detect cholesterol with this antibiotic that is Philippine, and we see here a blue signal, mostly coming from the surface. When we deplete the cells of esterols, we don't see the signal, and then we replace cholesterol in form of the dextrin, we see again cholesterol. And then, as I mentioned before, we also can test that this assay is working, checking for some of the enzymes that regulate de novo synthesis. As you can see here, the reductase, the famous enzyme target of statins that is given to people with hypercholesterolemia, is highly upregulated when cells are depleted of esterols, but as soon we add back cholesterol, it's rapidly degraded because it's no longer needed. The same happened with the other rate-limiting enzyme, SQL. So in these settings, we went to first experiment to check the FAMB levels. And so it's a first good indication what is going on with the ER-Phagy, because when these proteins, when ER-Phagy is activated, usually these proteins are also degraded with the process. So what's very interesting, we have these HeLa cells that express both isoforms, the long and the short isoform. And what's very interesting to see that the short isoform is accumulated when we perform depletion of esterol, and then is degraded in a cholesterol-dependent manner. So, and here also we see that the inducible cell line of HA tag FAMB2, the short one, is also degraded upon cholesterol addition after the accumulation when we perform sterol depletion. So what's very interesting to see here that the FAMB short undergoes cholesterol-dependent degradation similar to the cholesterol-related enzymes. And then we were we tested if this degradation of the FAMB2 short, which we see here that, upon cholesterol, is dependent on autophagy. And for that, we use a mutant of the short FAM that doesn't have the Lear motif, so cannot be targeted to autophagy. And this mutant is no longer degraded upon cholesterol. So this is a good indication that actually cholesterol is inducing the degradation of FAM via autophagy, and this is a good suggestion that actually cholesterol is inducing ER-Phagy. So we went to corroborate this, and for this we use this very well established ER-Phagy reporter, and this reporter is the FAMB itself fused in tandem with cherry and GFP. So when this construct in the cell is located in the ER or autophagosome, is yellow, and as soon it reach lysosomes, becomes only red. So we see a red dot basically. This is what we see here. In this condition, this is cells incubated in normal FBS. We see many red dots indicating that the protein goes from the ER to the lysosome. This is what we call the flux. So the flux is active. But as soon we perform esterol depletion using the strategy that I mentioned before, we see less of these red dots, and we could see also accumulation of the yellow signal in the ER. And importantly, when we supplemented back cholesterol here, 13 micromolar, we reestablished the amount of red dots. And this is quantified here. So this meaning, again, that in this ER-Phagy flux assay, cholesterol is inducing ER-Phagy. And nicely, we can see that opposite its depletion inhibits ER-Phagy. And this is what I was mentioning about the accumulation of the construct in the ER is important because it's indicating that cholesterol is required for early steps of the process. Because, for example, using the commonly lysosomal inhibitor bufferamycin, we accumulate these constructs in autophagous lysosomes, which is not the same for the esterol depletion. We use another ER-Phagy reporter now, a luminal construct that is the same principle, yellow in the ER and autophagosomes and only red in the lysosome. And when we deplete the cells of esterol, we see much less red dots, and then these start to be, we can detect them, again, these red dots, when we add back, supplement back cholesterol in a concentration-dependent manner. So again, cholesterol is inducing the process. And as I was mentioning before, we can induce, for example, ER-Phagy with taurine 1. And a good so here, for example, in the normal media, we see degradation of FAM with taurine 1. And this is impaired when we incubate the cells with esterol depletion media. Again, indicating that cholesterol is required for ER-Phagy. So since we publish that ubiquitination of FAM-B is required for the process, we wanted to see whether cholesterol is regulating this post-translational modification. We published that ubiquitination happens on the reticulum homology domain, and this stabilizes the clusters required for the process. And it was nice to see here in this ubiquitination assays that in this part of the Western blot, we see ubiquitinated enriched proteins, and then we plot against FAM, and we could see nicely that cholesterol induces ubiquitination of FAM. Here in a concentration-dependent manner, and here in a time-dependent manner, fixing 120 micromolar of cholesterol after depletion. So this happened for the long isoform. And we also tested the short isoform, it's ubiquitination, and it's the same. And here we can, in this part of the plot, we see that actually ubiquitinated levels of the FAM decreases upon depletion of esterol, and then it's rescue again when we add back supplemented back cholesterol for 2 hours. Then ubiquitination goes back because it's goes down because it's degraded. So, and then, of course, we wanted to see localization of, in this case, of FAM-B short in the cell upon depletion and supplementation of cholesterol. But it was nice to see that that depletion accumulates the protein in the ER, and we see much less colocalization with LC3 in comparison with the cells incubated in FBS, and was really striking to see a massive fragmentation of the ER when we supplemented 18 micromolar of cholesterol here. And many of these dots were positive to LC3. So again, suggesting that cholesterol induces ER-Phagy. And this is the summary of the first part. So I was wanting to say that clustering events of FAM are enriched in ER enzymes involving cholesterol biosynthesis, the novel one. That cholesterol is inducing ER-Phagy by, for example, inducing its ubiquitination and clustering in the ER. And the clustering we are testing now using a single molecule microscopy. And also we are coupling this with some mass spectrometry assays. So since this data is indicating that cholesterol is a positive regulator of ER-Phagy, we wanted to see how the cell proteome and also more focus in the ER proteome responds to changes in cholesterol levels. So, and then of course we wanted to see if ER-Phagy is involved in those changes. But first, to answer the second question, we performed some proteomics analysis in our cells that were incubated with FBS, normal FBS, with lipid depletion media, or with lipid depletion media that we supplemented back cholesterol with the dextrin. So we did this using TMT labeling, which allows quantitative proteomics. So this is, we got many data, but I will just show one analysis. So we have this volcano plot, and to the left is what is, we did a comparison between lipid depletion, and then when and in a condition when we have exogenous cholesterol with the dextrin. So here to the left is what is degraded upon cholesterol, and to the right is what is upregulated in response to cholesterol. So what was very interesting to see that the top hit that is downregulated is the reductase. So this is kind of our positive control because this is well known, well described. And but most of the proteins that were degraded upon cholesterol are ER membrane proteins and also Golgi membrane protein. This was very interesting. This is analyzed here in the geo terms and geo biological processes. So of course, in the biological processes, many of these enzymes degraded upon cholesterol are related to lipid metabolism, sphingolipids, ceramide, and many others. So, and interestingly, many core autophagy machinery was upregulated upon cholesterol. So it's nice to see that actually this correlates quite nice with the idea that cholesterol induces ER-Phagy, which means degradation of ER, which we see here. And for this, it is needed some autophagy machinery going up. And what was interesting that several proteins here that are downregulated upon cholesterol are also interactors of FAM134B, which raises the question that perhaps some of these proteins are degraded upon cholesterol in a FAM-B dependent manner. And this is what we wanted to address next. If ER-Phagy has some role in this cholesterol-dependent degradation of ER components. And for that, we did, again, the same proteomic analysis, but now including FAM-B knockout cells. So here we did the same, but we have double, we did TMT 18-plex. So here we are still analyzing the data. We got a lot of interesting things, but the most interesting thing was that here there is a correlation of what was degraded in the wild-type cell here in the x-axis, and in the y-axis is what is degraded in the knockout cells. And we could see that, for example, the reductase its degradation by cholesterol is not affected by the knockout of FAM, but there is a group of proteins, ER proteins, that its degradation apparently depends on FAM-B expression. Its degradation in response to cholesterol apparently depends on FAM-B. And this is also a graph here in this heat map. The purple means that the protein is less degraded in the FAM-B KO condition upon cholesterol. And so this was nice to see that upon cholesterol, there is a degradation that is independent on FAM, and there is a second group which we are more interested is a degradation of proteins in the ER that upon cholesterol that apparently depends on ER-Phagy driven by FAM-B. And just here as an example, we validated some of these proteins that I just showed you. For example, this MSMO1, which is also involved in de novo synthesis of cholesterol. So here, for example, in the control cell line, we accumulate this protein upon esterol depletion, but it's degraded upon cholesterol. Now, in the knockout condition, this degradation is not happening anymore upon cholesterol addition for this MSMO1. For the reductase, reductase is degraded independently of FAMB. And what was nice to see that actually this MSMO1 upon cholesterol treatment is able to colocalize here with LC3 vesicles that are also positive to FAMB. So indeed, we're going to a direction here that ER-Phagy participates in cholesterol-mediated degradation of ER and might be part of this feedback mechanism that I was presenting in one of the first slides. So now what we are trying to develop a bit more is that ER-Phagy is ER cholesterol sensing mechanism. So when there is too much cholesterol in the cell, ER-Phagy is activated, trying to adapt the cell to the stress that is caused by overload of cholesterol. So an important point that we are trying to study more in deep is how cholesterol regulates ER-Phagy. So for that, of course, we asked if this protein is a cholesterol binding protein. And FAM has been described that has several potential putative cholesterol binding sites here in the first hairpin, and also might be another putative cholesterol binding sites in the and one of the amphipathic helices. That is shown here in the amphipathic helices right after the second hairpin. So we tested this possibility. So we tested if cholesterol binds FAMB in cells. And for that, we use an analog of cholesterol, which is a clickable photoreactive cholesterol probe. We use this one. And this one here, this cholesterol has here a photoactivatable diacetyl group. And also here has a group that allows click chemistry. So it allows us to label this cholesterol with, for example, biotin or some fluorescent dye. So what we did is that we incubate cells expressing in an inducible manner FAMB. FAMB is tagged, so allow us to purification in a next step. So we incubate the cells with the cholesterol analog. Then we incubate the cells with UV light, so allows the cross-linking of this cholesterol to whatever protein is bound in the cell. So we cross-link the binding. Then after that, we perform cell lysis, normal cell one, and cell lysis. Then we purify FAMB, and then in pure FAM, we perform click chemistry to add this fluorescent dye to the cholesterol that potentially is bound to FAMB. So here is just to show that we can purify these proteins quite decently from mammalian cells. So it's we can do the assay that I just mentioned, and here is the result. So basically, we could detect cholesterol bound here to FAM. Here we can see the signal coming from the red fluorescent dye that is attached to the cholesterol, and this is we can observe much better the interaction of cholesterol with FAM in upon UV light. And this happened the same for the long isoform of FAMB and also for the short isoform of FAMB. So both isoforms are able to bind cholesterol. And we think that this binding is happening in the ER. And we are using this approach also to map interaction sites between FAMB and cholesterol. This will allow us if these putative cholesterol binding sites are indeed that, or validate those. And what's nicely also we can show that when we incubate cells here expressing cherry FAM-B, and we treat these cells with fluorescently tagged cholesterol here, and we can see here many vesicles positive to cholesterol and FAM. So this really indicates that cholesterol that rich ER binds FAM, then cholesterol might be inducing membrane helping the membrane curvature driven by FAM, and then this this piece of the ER is sequestered by autophagy. So autophagosomes coming from the ER positive to FAM, they might be also rich in cholesterol. And since the question still is how cholesterol is inducing ER-Phagy, we are trying more biophysical experiments, and we have an assay here in the lab that we incubate FAM-B, pure FAM-B with liposomes, and we don't know how exactly this is happening, but when this incubation happens, we convert big liposomes in very little ones. So this is what we call that FAM remodelates liposomes. So what we did here to test how cholesterol is doing is that we have FAM-B generating this incubated with liposome without cholesterol. Here FAM-B incubated with liposome with cholesterol. And we could see here in these three images that the liposomes that are contained cholesterol are much smaller than the ones that are generated when FAM is incubated with liposome without cholesterol. And this is a suggestion that FAM-B is able to remodel membranes much more efficiently when cholesterol is there. And this can be also extrapolated to what is happening in the cell. So this is our model of study. So we think that specific sites in the ER where ER-Phagy is about to happen, these FAM-B clusters recruit cholesterol biosynthesis related enzymes which can generate cholesterol. But also cholesterol can be incorporated to these sites by intracellular transport that is also positive. And this cholesterol binds FAM-B, and this helps to deform the membrane, which then will be sequestered by the autophagosome. And then also this induces other signals like the ubiquitination of the FAM in these clustering events. And what we are trying to develop, what I was trying to say to you is that we are trying to say that this is a cholesterol sensing mechanism. So basically it's activated when there is too much cholesterol in the cell. And how this is happening is that cholesterol is regulating biophysical properties of the membrane when FAM is there, helping to boost this deformation of membrane required for ER-Phagy. And the last part of the talk, I will be very brief. That is where we would like to apply this mechanism, and we would like to apply it in liver. And we observed similar things in hepatocyte cell models, which basically is that there is degradation of proteins ER proteins that depend on FAM. For example, this MSMO1 here is degraded upon cholesterol in control cells, but this is not happening when FAM-B and also another paralog is absent in the cell. So ER-Phagy also participate in cholesterol-mediated degradation in hepatocytes. We are now doing mice work with this. For example, here we were able to isolate primary hepatocytes, which, by the way, the main isoform that they express is the short one. And then, and we could see also that upon cholesterol incubation of this primary hepatocyte, we could see much more endogenous FAM-B with LC3 colocalization, indicating that also in primary hepatocyte, cholesterol is able to induce the process. So we are now trying to study this mechanism that we are elucidating in liver disease, specifically in MASH. So this disease is characterized by is triggered by metabolic dysfunction and insulin resistance, which leads to lipotoxicity. You see like a fat liver and full of lipid droplets. And then this leads to cellular stress, inflammation, and fibrosis, which at the end can lead to hepatocellular carcinoma and other more severe liver disease. And this is it's very related to obesity, which is a main problem in many parts of the world. So we have a specific knockout of FAM-B in the liver. We are creating several models about this. With diet, we are inducing fatty liver, and we are inducing MASH, and also MASH plus acute on chronic liver failure, which is kind of the late step of the whole liver disease. So with this, we want to study how the presence of FAM and ER-Phagy impacts the development of these different pathological conditions. We have some preliminary data about what we were expecting in our mice model of MASH. We see upregulation of many inflammation response upon MASH in the control animal, and there is a downregulation of cholesterol biosynthesis in the liver of the animal that is developing MASH. So our models are working and are indicating a potential link. It's known that cholesterol has a role in development of MASH in the liver, but this is ongoing. And we also see that in MASH, there is a tendency of downregulation of FAM-B and FAM-B2. So this is a good suggestion that something is going on with the pathway upon a pathological condition. And what is very interesting, and this is where this project is leading to, is that now we are able to identify FAM-B in the lipid droplets, and which is accumulation of lipid droplets is a hallmark of liver disease. So what is doing FAM there now in lipid droplets is a mystery so far. We are trying to figure it out, but is definitely indicating that FAM-B and potentially ER-Phagy is really connected to lipid metabolism. And as a good example that I just was talking is cholesterol metabolism, but other lipids can be involved in, for example, lipid quality control, which is a very interesting, relatively new topic that is accumulation of lipids that are oxidized, which can be also recognized maybe by this FAM-B. Some of the lipid droplets that contain FAM also recruit autophagy machinery, which is very interesting, and we are developing this part as well. So what I want to say is that we are studying how ER-Phagy regulates lipid metabolism. I just gave you one example about the connection of cholesterol and ER-Phagy, and then we are linking this fundamental processes to the pathophysiology of MASH. And we are also, of course, interested what happened, for example, with advanced liver disease, like, for example, in cancer. And with that, this is my acknowledgement. Many people is involved in this project. Many thanks to Ivan, which is my mentor, and I'm always grateful to him for the scientific direction and for the fantastic scientific network that we have here in Frankfurt. Adriana, which is she will be giving a talk also in 2 more weeks, which she will be talking about more interesting stuff about mitochondria contact sites. She is my forever partner, right? And we are developing together this project. Adriana is working in the mice part together with Anya and Christine. Matthias is working with the lipid droplet part with Peter. And V9 is working, we are working together with the biophysical part, and Rubina and Thorsten, they help us with the proteomic analysis. Also many thanks to Ashvin and in Heinemann's group, which is helping us with some super resolution microscopy. And in the lab of Ram and Sergio, they are helping us with some molecular dynamic simulations to try to model the interaction of FAM with cholesterol. Also, many thanks to Stefan Heima, which is helping us with the electron microscopy. And we also thanks to the Albert Pieper Lab and Daniel, that they are also providing us with some patient samples that we can evaluate some of these aspects in human samples. So thank you very much for your attention, and happy to answer some question if you have some.
Bueno, Alexis, muchas gracias. Tremendo trabajo que has realizado. Y yo voy a partir con la primera pregunta porque en realidad ahora comprende que la regulación del colesterol es bastante compleja. De hecho, muchas veces uno piensa que el colesterol es como un verdadero segundo mensajero, puesto que, por ejemplo, los niveles intracelulares dependen de la parte exógena de las lipoproteínas, pero también el hígado tiene la propiedad de sintetizarlo.
Sí.
Entonces, de
Ujú.
hecho, mi pregunta va a que el mismo colesterol regula la expresión del receptor, por ejemplo, DLL o la expresión de la HMG-CoA reductasa. Entonces, ¿cómo se logra censar este balance entre, por un lado, la síntesis de elementos que conducen a mayor colesterol y, por otro lado, de alguna forma, la degradación o expulsión dentro de la célula? Cómo se logra ese fino balance?
Claro, esa es la gran pregunta. Concuerdo mucho con eso. Por ahora se sabe mucho, por ejemplo, que la reductasa es degradada vía protosoma cuando hay una acumulación de colesterol en la célula. Ahora también hay toda una regulación a nivel de la captación de colesterol. Ahora hay unas nuevas
Sí,
moléculas, PSCK9, que
sí,
participa, y hay
PCP9,
una, sí, y
un
hay unas
inhibidor químico, de
importantes.
hecho,
Sí, los clinical trials están súper bien ahí, pero claro, nosotros estábamos más enfocados porque en el campo del ER, siempre se ha sabido que el ER es el mayor organelo sensor de colesterol, el intracelular. Y lo que este trabajo propone es que cuando mucho colesterol es transportado hacia el ER, y ER-Phagy va a ser activada por colesterol, pero a su vez con un mecanismo feedback que va a tratar de remover colesterol desde la célula. Y estas vesículas, estos autofagosomas que van del ER, que contienen FAM, podrían estar transportando el exceso de colesterol hacia el lisosoma. Y el lisosoma ahí, bueno, esa es otra historia, pero el lisosoma es otro
otro
major
mundo.
organelle
Sí.
que participa en el metabolismo del colesterol. Pero la idea,
Oye, pero
claro,
una de las cosas que tenía es, cuando tú hablas de retículo, estás hablando del liso, ¿no es cierto? Esto
estoy
ocurre en
hablando
el
del...
retículo liso,
Sí, eso es otra parte del proyecto que lo hemos estado elaborando. Es liso, es lo que hoy... El liso, claro, hoy día se habla más del tubular, del retículo
ya del tubo,
endoplásmico
los sheets,
tubular. Sí,
ya,
entonces
ya,
esto pasaría principalmente en el tubular ER, en el liso.
ya,
Y la
perfecto.
degradación del IAR es de IAR liso, digamos. Esa es y ahí es donde no se sabe muy bien en qué regiones del IAR liso ocurre este IAR falli. Y obviamente algunas técnicas como super resolución están ayudando un poco a entender un poco en más detalle la morfología del IAR, y ya están surgiendo algunos detalles. Ahora también en este mecanismo, bueno, hay muchos aspectos interesantes que hemos estado hablando, pero los contact sites también tienen mucho que ver, porque el colesterol que llega al IR viene principalmente de la superficie. Y ahí hay unas proteínas muy interesantes que transportan el colesterol desde la superficie al ER y nosotros hipotetizamos que también ese mecanismo podría estar involucrado en la activación de ER-Phagy por colesterol.
Ya, perfecto. Bueno, aquí hay más palabras levantadas. Héctor, adelante.
Yo quisiera consultarte algo porque si hay algún tipo de conexión entre el ER-Phagy con la activación del NLRP3 inflamasoma, ya que hace poco salió una publicación precisamente con estas proteínas que estabas hablando recién del Aster, que
Sí.
participan en la activación del NLRP3 inflamasoma. El colesterol aumenta la estabilidad del NLRP3 y facilita la
Ujú,
activación del NLRP3 inflammasoma, un poco al contrario de lo que está haciendo el ER-Phagy, que es la degradación de
ujú.
esta. Entonces, no sé si hay algún comentario respecto a esa conexión entre ambas vías, considerando también que en el RP3 inflamasoma está asociado a los MAM en el
Ujú.
retículo.
Sí, sí. Bueno, no estoy muy seguro acerca del inflamasoma, pero lo que, inclusive en este laboratorio, lo que se ha tratado de hacer es un poco linkear si ER-Phagy tiene algo que ver con la respuesta innate immunity. Ah, no, no, la verdad desconozco la respuesta para lo del inflamasoma, pero sí le puedo decir que FAM aparentemente activa STING pathway, que es que esta vía, claro, que es proinflamatoria. Pero, pero claro, eso ha sido publicado ya, hay varios, unos dos reportes acerca de eso, pero honestamente nosotros no lo hemos podido reproducir tal cual.
Ok.
Sí, gracias.
¿Más preguntas? Oye, así como existe la comunicación retículo mitocondria, existe en este caso, porque existe un ejemplo de la comunicación retículo-membrana plasmática. Cuando tú tienes que se depleta de calcio, está todo este sistema del ORAI, STIM-1, de que se vuelve a repletar de calcio. ¿Hay algo
Ujú.
así similar que ocurre con el colesterol, así como esta comunicación tan física, estrecha, en relación a lo que tú estás estudiando?
Sí, sí, Héctor estaba preguntando eso y él mencionó estas proteínas que se llaman Aster o Gram D1.
¿Ya?
Y estas proteínas estarían cuando hay un exceso de colesterol en la superficie, estas proteínas transportan el colesterol accesible de la superficie hacia el IR. Bueno, aparte yo mostré muchos modelos en que juego con el colesterol, lo depleto, lo coloco de nuevo, pero de hecho, por ejemplo, generamos ya unas líneas que son Aster Gram knockouts. So, esperamos que en esa célula en el cual hay menos transporte de colesterol desde la superficie al IR, y el falle debería estar afectada negativamente en ese concepto. Esa es una conexión importante entre los contactos que hay entre la superficie y el IR con respecto a colesterol. Y por supuesto lo que tú estabas mencionando de todo lo que tiene que ver el calcio.
Ok. ¿Y más preguntas? ¿No?
Tengo
Bueno,
una, yo sé que.
Alexis. Sí,
Espera.
Mario.
Hola Alexis, muchas gracias por tu presentación. Tenía la duda si esta misma familia de proteínas, la FAM de la 134, participan también en la autofagia de Golgi.
Esa es una interesante pregunta. La respuesta eh, es no, no. Bueno, hay eh, dos, tres, cuatro artículos en los cuales han descrito Golgi Phagy receptor más específicos que son pero lo que sí es que Golgi y ER se degradan, digamos, hay un muy bonito paper hace un par de años en que cuando uno depleta la célula de aminoácidos, que es el principal estímulo de ER-Phagy, también ellos ven que Golgi se activa. La degradación de Golgi se activa bajo el mismo estímulo que induce la degradación de ER. So, yo lo que sí le puedo decir es que ambos procesos podrían ser gatillados por los mismos estímulos. Ahora, la maquinaria eh, difiere un poco. Ahora, hay algunos reportes que localizan FAM en el Golgi. En mi experiencia, a veces lo puedo detectar en el Golgi. No sé por qué, pero la mayoría de las veces está en el IAR. Así que yo creo que em, por ahora yo diría que FAM solo participa en la agregación de IAR, no Golgi.
Gracias, Alex.
Ok, Clarita,
Hola,
Clara,
sí, sí,
sí.
sí. Hola, gracias Alexis por tu presentación, es muy interesante. Te quería preguntar respecto de la autofagia, ¿cierto? El retículo en condiciones de estrés recovery, ¿cierto? Cuando ya termina o el proceso de estrés ya, digamos, se activó la UPR y todo lo demás, ¿cierto? Y ya se recupera un poco la homeostasis del retículo. Se ha visto,
Ujú,
por ejemplo, que CEC62 es importante, ¿cierto?, para el reclutamiento y la inducción de la autofagia.
ujú.
¿Existe alguna, y eso ocurre en el retículo más bien en laminar, ¿cierto?, rugoso, más que en el tubular, como tú mencionaste, que es importante FAM. ¿Se
Ujú.
sabe algo respecto de si estos mecanismos podrían ser transversales o si estas proteínas como SEC62 también podrían estar en estas regiones ricas en colesterol? ¿Hay alguna información respecto de eso? Gracias.
Em, sí, buena pregunta. Sí, hay muchos reportes tienden a separar un poco este receptor, participa en la degradación del rugoso o las chits, ¿verdad? Este receptor participa en la degradación de los túbulos, el liso, pero la verdad, en mi experiencia, muchos de estos receptores trabajan juntos. Em, por ejemplo, durante estos años hemos hecho muchos interactomas, muchos proteómicas de los FAM y usualmente detectamos SEC62, detectamos los otros FAM, sí, sí, especialmente detectamos TEX. El que tiene un poco de otra función es el CCPG, el otro receptor. So, por lo tanto, mi respuesta es que sexo sentido sí podría estar involucrado en este proceso también. Em, no lo... a ver, no lo he medido, pero sí, pienso que sí debe estar también, debe ser parte de la maquinaria. Y porque, por ejemplo, el FAM, en el artículo original, hace ya 10 años, se propuso que mediaba la degradación de las chits, del rugoso. Pero, pero también en mi experiencia y la de otros colegas, también lo localizamos mucho en los túbulos. Por lo tanto, y para mí, participa más en la degradación de túbulos que en la de las chits.
O sea, que tiene lógica por la estructura y que se inserta en la membrana que favorece la formación de la curvatura, que
Claro.
por eso puede estar más presente en el túbulo que en las láminas.
Sí, de hecho yo tengo bueno, no he hecho nunca una microscopía más avanzada que la confocal y alguna otra, pero FAM tiene una tendencia mayoritaria a localizarse mayormente en túbulos. Ahora, más en túbulos que en chits. También uno los localiza en las chits. Em, pero hay algunos artículos que también sugieren que ER-Phagy ocurre más específicamente en los túbulos que en las chits. Y En el caso de ahora que en este trabajo hemos jugado harto con el colesterol y la síntesis de colesterol y de otros lípidos ocurre mayoritariamente en el liso, los túbulos. Como Sergio preguntaba, yo creo que ahora en este caso estaríamos hablando de una ER-Phagy que ocurre específicamente en los túbulos.
Perdón, sería diferente a la que ocurre cuando se tiene que dar cuenta de la membrana sobrante después de que termina la UPR, ¿o no? Porque esa es
Eh,
membrana, digamos, es más lámina que túbulo, ¿o me equivoco?
em, ¿Me repite, por favor?
Porque, porque, porque lo que yo tengo entendido, ¿cierto? Una vez que en este paper que salió, no me acuerdo ya qué año, cuando
Ujú.
menciona, ¿cierto? Este mecanismo como de stress recovery, cuando describen a SEC62, ¿cierto?
Sí,
Dicen
sí,
que es
sí.
muy importante para un poco favorecer la degradación de toda la maquinaria que se sobreexpresó durante la UPR, de la
Mm,
chaperona, sobrante, ¿cierto? Un montón de otras proteínas
ajá.
y también de la membrana. Entonces ahí yo entendería que aumenta la autofagia, ¿cierto? De toda esta membrana y todo este contenido que ya no se requiere una vez que la UPR resuelve, ¿cierto? El
Ujú. Sí,
estrés.
sí, sí.
Entonces, por
Ujú.
eso, en realidad, es más que nada eso, si es que había alguna información respecto de, no sé, que hubiera enriquecimiento de colesterol y eso favoreciera la
Mm.
autofagia de esa membrana. O en este caso también si se entiende como mecanismos distintos, se le llama también autofagia o ER-Phagy también a ese proceso, ¿no?
Sí, le llaman ER-Phagy Recovery, algo así. Eh, sí, lo que le puedo decir es que hay unos grupos, de hecho acá en Frankfurt, que están haciendo cryo-EM tomography. Entonces están ya llegando a un nivel, claro, de microscopía electrónica y están investigando cómo luce el ER dentro de los autofagosomas. Em, Ahora, claro, sería muy interesante. Yo creo que dependiendo del estímulo, del tipo de estrés, el contenido del IAR y la estructura del IAR va a ser diferente de la cual el IAR que se recluta al autofagosoma para degradarse. En el caso del SEC62, no estoy muy seguro, pero em, eh, para mi entendimiento hay en este grupo que está estudiando la ultraestructura de IAR en el autofagosoma. Si bien recuerdo, hay unos IAR que están muy asociados con ribosoma, que vendría siendo el rugoso, ¿verdad? Pero hay otros IAR que no tienen ribosoma, por lo tanto ese vendría siendo más liso. Así que, claro, dependiendo mucho del tipo de estrés, porque por ejemplo, em, se requerirá, por ejemplo, que el IAR que se degrade va a degradar, por ejemplo, una fracción menor de los ribosomas que están asociados al IAR y, como usted decía, por ejemplo, involucrados en respuesta a la UPR. Pero en el caso del colesterol, no. De hecho, quería mostrar esa data porque en estas proteómicas que hicimos de cosas que se degradan en respuesta al colesterol, veo muchas proteínas ribosomales. Cosa que, claro, me genera un poco de ruido y no sé cómo interpretar todavía. Pero claro, la interpretación podría ser que estos parches que te estoy mostrando aquí en este modelo, también, por ejemplo, podrían tener incorporados muchos ribosomas que estaban sintetizando, que están sintetizando, por ejemplo, estas enzimas que participan en la de nuevo biosíntesis o chaperonas que están lidiando con la sobrecarga de colesterol en el hígado. Eh, pero, claro, su pregunta es muy buena porque y se está tratando de responder con esta nueva tecnología, bueno, no tan nueva, pero ya la gente la está haciendo más comúnmente, que es esta cryo-EM tomography. Ya ahora tienen unos autofagosomas hermosos con el contenido de IAR dentro del autofagosoma, pueden hacer mediciones, incluso, bueno, La meta de ese tipo de estudio es identificar las estructuras de las proteínas que están ahí. Bueno, en 2 semanas más, Adriana les voy a mostrar esa tecnología. Ella se asoció con estos grupos y les voy a mostrar algunas de estas Cryo Tomography con contenido dentro de los autofocus.
Gracias.
Está muy interesante la conversación, pero ya estamos en la hora, así que agradecemos tu interesante seminario, así que los quedamos cordialmente invitados para en 2 semanas más volver a reunir. Muchas gracias y buen fin de semana allá en Frankfurt.
Ok,
Chao.
muchas gracias. Saludos.
Muchas gracias, Alexis.
Gracias por la organización. Chao, chao.